Inflammatory trigger a new clue in Alzheimer's and PSP. Featured Neurology Neuroscience, January 6, 2023. Summary: an inflammatory trigger, like one present during viral infections, is elevated in those with Alzheimer's disease and progressive supranuclear palsy, PSP. Source: UT San Antonio. Scientists from the University of Texas Health Science Center at San Antonio, UT Health San Antonio, today reported that an inflammatory trigger, like one present during viral infections, is elevated in Alzheimer's disease and progressive supranuclear palsy, a rare brain disorder. "We have identified a new trigger of brain inflammation in these disorders," said Elizabeth Ochoa, study author from UT Health San Antonio. "The finding, published in Science Advances, is novel for this reason," she said. Ochoa, a recent doctoral graduate, and her mentor, Bess Frost, Ph.D., study senior author, are investigators with the Salmon and Barshop Institute for Longevity and Aging Studies, the Glenn Biggs Institute for Alzheimer's and Neurodegenerative Diseases, and the Department of Cell Systems and Anatomy at UT Health San Antonio. Frost is the Bartell Zachry Distinguished Professor for Research in Neurodegenerative Disorders. Alzheimer's disease and progressive supranuclear palsy are marked by toxic deposits of a protein called Tau. Their research found that Tau-induced jumping genes, which can relocate or copy themselves to other locations in the genome, form double-stranded RNA. This abnormal RNA mimics the inflammatory trigger that is also present in viral infections. Transposable elements, the so-called jumping genes, are a new area of interest in understanding Alzheimer's disease. Our study provides new insights into how they can drive the disease process in addition to their ability to jump, Ochoa said. These double-stranded RNAs look like a virus to the immune system, even though the jumping genes are a part of our normal genome. This shows a brain. Alzheimer's disease and progressive supranuclear palsy are marked by toxic deposits of a protein called Tau. Images in the public domain. The researchers detected accumulation of double-stranded RNA in postmortem brain tissue from patients with Alzheimer's disease and progressive supranuclear palsy, and in brains of mouse and fruit fly models of tauopathy. We found substantial deposits of double-stranded RNA in astrocytes, which are cells that provide metabolic support for neurons, regulate neurotransmitters, and maintain blood-brain barrier integrity, Frost said. In aging and disease, astrocytes respond to injury and disruption of the neuronal environment. Our findings open new doors for understanding astrocyte biology and their role in transposable element control. Loss of neurons, which are cells of the central nervous system, is progressive in Alzheimer's and other neurodegenerative diseases.